Okay, so this video talks about Chapter 5, the first part of Chapter 5 actually. And Chapter 5 is entitled Mostly Microorganisms. So since most microorganisms are found in the first few kingdoms, so in Kingdom Bacteria, Kingdom Archaea, and Kingdom Protista, this chapter covers those, as well as Kingdom Fungi, because Kingdom Fungi has one type of unicellular microscopic organism. So we will start with the first of those kingdoms, which is Kingdom Bacteria. So this video is going to cover Kingdom Bacteria and Kingdom Archaea, and if you recall, they are both prokaryotic, so their cells are very simple, they're very tiny, very small, and we'll look at each of the kingdoms a little more closely so that you can understand the differences between them. So Kingdom Bacteria, these are a few of their features mentioned already. They are prokaryotes. They include prokaryotes. They reproduce asexually usually, but under certain conditions they may reproduce using conjugation, which is kind of like sexual reproduction in that there's some changes in their genetic information that allows them to adapt in certain ways. So bacteria are really, really tiny, and they can live in very, very unique and very like almost endless types of environments. Those in Kingdom Bacteria, or Kingdom Eubacteria, as you might see in some textbooks or on some sites, they are not as able to live in very extreme environments like what you'll see with Kingdom Archaea, but they can live in like a lot of different places. So one thing that you might be surprised with, though, is the fact that bacterial cells, we have more bacterial cells than human cells on our bodies. So some of them are on our skin, some of them are inside of us, in our digestive tracts. It's kind of crazy that there's more bacterial cells than human cells. The other point that I want you to understand is not all bacteria are harmful. In fact, 99% of the bacterial population are helpful. And we're going to look a little more closely at some of the things that bacteria can do for us, but here's a couple. They're used to make yogurt, cheese, medications like insulin, and other products that we use. So we can engineer them to do what we would like in terms of building things. So they're a lot more helpful than you would think. One of the objectives for this section is to understand how bacteria are classified, and there's a few different ways, a few different categories that they use to classify them. So the first is classifying them by shape. So there are three main types of shapes for bacteria. The first is called cocci. Cocci is plural, or coccus singular, and basically they are round. So these pink ones would be cocci. Bacilli, bacillus singular, are rod-shaped. So E. coli would be a good example of a bacillus type bacteria. And spirillus singular, kind of like the name suggests, they are spiral shaped. So here's some spirillii bacteria. Not only are they categorized on their shape in general, but also they're categorized in how they might be arranged in their living. So sometimes they actually form little groups, and depending on the type of connection they have, like how they are grouped together, they might have another part to their name. So if you see the prefix diplo, that diplo means they're in pairs. Di is for two, so you'd have them stuck together in pairs. Strepto, like in strep throat, streptococcus, they are arranged in chains, so they're like in a straight line. Staphylo means they're in clusters, so kind of like a bunch of grapes, or a bunch, well, not always round in shape, but that they're stuck together in that sense. Another way that they tend to be classified is based on their cell wall structure, and they actually have a series of stains that they'll use if they're working in a microbiology lab. Like if you take microbiology at university, for instance, then they would get you to perform this particular staining, which is called gram staining, but they'd also have you do some other types of staining to deduce exactly what kind of bacteria might be there. Gram staining quickly indicates what kind of cell wall they have, and so the stain itself is purple, and if the bacteria has a certain kind of cell wall, they'll hold the stain inside of them and they'll stay purple. So we call them gram-positive bacteria. So they have a thick cell wall composed of peptidoglycan, and when the stain is introduced to them, they retain the stain. The stain stays in them. Gram-negative bacteria, they have two cell walls, one of which, the inner one, is really thin, peptidoglycan layer. They don't retain the purple, so because of the difference in their cell wall, the purple stain can't stay on them, so they would appear pink instead. Okay, and these are just some examples of bacteria that are gram-positive. So Bacillus anthracis is anthrax bacteria, and they are gram-positive, so they would retain the purple color. E. coli are gram-negative, so they would not. So here is a typical type of bacteria that you'd find in your mouth. So if you look at it, okay, they are found in chains. So if they're found in chains, if you remember, they're found in a straight line of chains, then that would be strepto. Okay, so this is based on whether or not they, how they're grouped, how they exist in groups. So the blue is classifying them based on their shape, but how they sit in groups. The green is based on their actual shape. So individually, what shape are they? And they are round, so we would say Streptococcus mutans is its name, and it is the most typical one found in your mouth. MRSA, on the other hand, if you go to the hospital and you have to go to the emergency area, or the outpatients, as we call it, then they will, one of the questions they ask you is, have you been told that you have MRSA? And that is the, basically it's a superbug. So they know by you telling them that you have carried these bacteria that are really resistant to antibiotics. So they would have to treat you with special antibiotics to try and get rid of infections that you might have, that sort of thing. So it tells them a lot, that question. Okay, so let's take a look. Now these are stuck together in bunches. So they're not just in twos, and they're not in straight lines, so they must be staphylo. And again, they are round in shape, so we'd say Staphylococcus, and they are Staphylococcus aureus, and Staph aureus is a very common bacteria, They're commonly found on your skin, but they can lead to infections and illness if they find a way to get inside of you and to colonize in higher numbers than they would normally be found. Okay, so that's Staphylococcus aureus. And so the MR in MRSA stands for methicillin resistant, and SA is Staphylococcus aureus. And they're both purplish. They retain the purple inside of them, so they would be gram-positive. Okay, so that was classification by shape. Now we're going to look at classification by nutrition, and this is just kind of a schematic diagram, which is just words and arrows that might hopefully help you keep them straight. So, if their major energy source is light, then they could be autotrophic or heterotrophic. If they are heterotrophic, then we would call them photoheterotrophs. If they are autotrophic, they would call them photoautotrophs. If their major energy source instead is inorganic compounds, then we would call them chemoautotrophs or chemoheterotrophs. That's where the name part comes from. This is just to summarize, okay? So, photoautotrophs use light energy to convert carbon dioxide into molecules that can be of use for the cell. So, photoautotrophs would include a lot of bacteria, but it would also include plants. Okay, so plants use light energy to convert carbon dioxide into molecules that can be of use for the cell. Photoheterotrophs use light energy to convert organic molecules from one form to another. So they would use light energy to convert food molecules that might already be inside of them to another form that can be used in the cell. So they still use light, but they have to eat other things to get their food stuff. Now, chemoautotrophs, instead of undergoing photosynthesis to make food stuff, they would undergo chemosynthesis. So they would use inorganic chemicals like sulfur and things. Maybe an example would be certain bacteria that are found near the, especially Archaea bacteria that are found near the vents at the bottom of the ocean. They're super, super hot. And there's inorganic chemicals like sulfur-based chemicals that they can use instead of light to get energy. And so they can make food stuff using the inorganic chemicals. Chemoheterotrophs derive their energy from organic chemicals that are acquired from the environment. So we would be a type of chemoheterotroph. So like with the viruses, I gave a handout about the lytic and the lysogenic cycles for reproduction. Remember, they can't reproduce on their own, and they have to have a host. I'd like you to be able to sketch and describe, explain the types of reproduction found for bacteria. So asexually, they reproduce using what's called binary fission. So bi means two, fission means to split. So basically, the cell is going to elongate, make a copy of its DNA, and then form a wall that divides it into two new cells. Okay? Because the DNA is copied directly, it's exactly the same, so it's like you're making clones of the first bacteria. Okay? It happens very, very quickly. The different bacteria reproduce at different rates. In math class, you'll probably be learning about bacterial growth and when you're learning about exponential functions and logarithms and things like that. But basically it's binary fission, so the population would, oh, my video's not working, would double every time. With each reproduction, the number of bacterial cells would double with each time frame for doubling. So it would be a doubling exponential function in math. So here's another picture. Okay, this one is more word description of what happens. This is a picture showing you what happens. So the cell basically is going to get longer. It elongates, gets bigger. There's the DNA. Remember, they don't have a nucleus. So they make a copy of their DNA, or replicate it, and then a cross wall, or a septum, will form between them to divide them into two. So it completely separates them, and then they will separate into two bacteria that are exactly the same as the parent bacteria. So they're genetically the same. Everything's the same about them. Hopefully this, goodness. I have to update my links. Okay, this one does work. So this one is 440 times normal speed, but it'll show you binary fission. So it elongates. You can't really see the DNA replicating inside, but that's what happens, and they form a wall, and then they have new cells. And this is what happens when things like E. coli colonize your body. And there you go. The other way that they reproduce, and it only happens at certain times, is through conjugation. And in conjugation, usually only happens when things are unfavorable, or when the bacteria want to share certain properties that allow them to maybe resist antibiotics or to adapt to the environment. So basically, this is just a slide that explains it in words. Basically, this would be the donor bacterial cell. Okay, sometimes we'll just call them a positive or positive strain, because it has a plasmid, which is, here's the chromosomal DNA, but plasmid DNA is a circular piece of DNA, and they often have on them a gene that codes for something that helps the bacteria to adapt to their environment, or to make gives them resistance to certain antibiotics, for instance. Okay, so the one that has that plasmid would grow or produce this long extension called a sex pilus. So they have, remember we learned about prokaryotic cells, and they have pili, or things that almost look like cilia, around the outside of them normally, so they can stick to each other or stick to their host cells. But this one is a super long one, and it's adapted for a special function. So we call it the sex pilus, and its job is to attach to a recipient bacterial cell, or a negative strain, what we would call it, because it doesn't have that plasmid. Okay, so the sex pilus will allow it to attach to a recipient cell, and it kind of allows them to become closer together, and then the donor cell will make a copy of its plasmid DNA and send a copy through to the recipient cell. The two cells will separate, and now each of them would have the plasmid that gives them resistance. Okay? And each of them can go and be donors to other cells. So this is a simple animation. Now sometimes what the plasmid might code for is a special resistance marker that would appear on the outside of the bacterial cell. Okay, so we're going to watch. I'm just going to pause it here. Okay, so this would be the positive strain. You see it has the plasmid inside, and these are its markers. Okay, so it has certain things on its cell wall, on its surface, that are giving it resistance at this point to something in the environment. Maybe it's to a certain antibiotic. Okay, so this one is going to form, remember, the sex pilus. So let's see. Forms the sex pilus.
Makes something called a pilus and transfer the gene over. Boom, it encodes this thing. Now we have an antibiotic resistant gene in.
Oh, so did you see that? Remember back here, it didn't have those markers, but as soon as it gets the plasmid DNA, same gene, but it's the plasmid DNA with a certain gene on it.
Make something called a pilus and transfer the gene over. Boom.
Oh, there they come. See, they're being produced because of the gene that's on there.
In here, and this organ now has the resistance to the antibiotic.
So now this one would also be considered a donor or positive strain, and it could go and do the same thing to another. So that is binary fission. Okay, so binary fission is their normal means of reproduction. Okay, it's asexual. Asexual simply means that the parent cell will produce two new cells that are identical genetically to the parent cell. There's no changing of genes, there's no exchange of genetic material that would allow them to adapt to their environment. Conjugation is kind of a form of sexual reproduction, in that a sex pilus forms and some genetic information is transferred, but normally in normal sexual reproduction, the genetic information is exchanged between parents or combined in a new manner. So some would say this isn't true sexual reproduction, but it's not asexual, and it does give it new features that can allow it to adapt. So I'm kind of on the fence with that one. So we'll still categorize it as sexual reproduction like they used to. Okay, so here is a representative for kingdom bacteria, and they are the cyanobacteria, or blue-green algae. They're found in phylum Cyanobacteria, and they're probably the first organisms on Earth to use photosynthesis. They're really, really important producers in the aquatic environment. So when we talk about algae, and we talk about the algae that floats on the top of water bodies, then you would find a lot of blue-green algae in there, and they are bacteria. How are they helpful? So we mentioned a couple of these facts before, but they can be used to make foodstuff for us, like yogurt and cheese and butter and even vinegar. They can be genetically engineered using plasmids to be like mini factories where they mass produce proteins, and then we can harvest them. So this is what we've done in order to make insulin. So we take the human gene for insulin and put it on the plasmid, and then introduce the plasmid into the bacterial cells, and then the bacteria do all the work for us. They use their machinery and make these proteins. So they make a whole lot of insulin, and then we just harvest it and make sure it's isolated and cleaned up and put into bottles. They are also decomposers, so that means that they break down decaying matter, recycle the nutrients, and they also fix nitrogen to make our soil fertile and provide nitrogen for other organisms. So there's a lot of bacteria that tend to be associated with little roots on plants to help fix nitrogen and help make the soil fertile. They can be found in stomachs of many herbivores, especially cows and other ruminants. It helps them digest and make use of cellulose that's in the plant cell wall, so that's why they tend to eat grass and we don't. They also live in our intestines, in our large intestine, and they help us to make vitamins. So we have some good bacteria in us all the time. Now, antibiotics. We should have a bit of an understanding what we mean by antibiotics. Basically, they're selective poisons, and they usually will kill the desired bacteria. Not that we desire to have them, but that we desire that they're the ones that we want to have killed, but not affect the cells in your body, usually. That's the intent of them, but sometimes there are some side effects. Different types of antibiotics affect different bacteria in different ways. So one type of antibiotic might work for this type of bacteria, but not for another. Okay. They might inhibit their ability to turn glucose into energy, or inhibit their ability to construct their cell wall, or make it strong. Those are some examples of what they can do. Okay, so this is showing penicillin. Penicillin interferes with the cell wall production, and so it weakens the cell wall so that they can easily be destroyed by the host cell, by the human cells that have these in them. Either way, the bacteria dies instead of reproducing. So this is just a little diagram showing these are all types of antibiotics, classes of antibiotics that would interfere with cell wall production. Okay, and these are ones that would interfere with production of necessary proteins, like enzymes and other things inside of the bacteria. And then these types are ones that interfere with DNA replication and repair in the bacteria. So those are just some classes of antibiotics and how they tend to have their effect. There's some little videos there. I'll link them onto the website, and then you can watch them at your own desires. So antibiotic resistance. It's one thing to take the antibiotics, but the scary thing is when you develop antibiotic resistance. So the main reason for antibiotic resistance coming about is that physicians tend to prescribe antibiotics a lot more, and for things that may not actually respond to antibiotics. So if you have a flu or a cold, then those are things that aren't really going to be affected by antibiotics, but yet we still tend to run to the doctor, and then the doctors will sometimes prescribe them, and then they're taking medications that aren't really working against what they're intended to work against, and it ends up causing any bacteria that are there to develop resistance to those antibiotics, so that when you are sick with those bacteria, then the antibiotic won't work. So how do these bacteria end up developing resistance to the antibiotics? Well, like we saw, they might share plasmids that offer resistance, so they may undergo conjugation and share them. This is a diagram kind of showing some of the ways that a bacteria might develop resistance. So maybe the plasmid causes the bacteria to be able to make a pump that pumps the antibiotics back out. So here's the antibiotic coming in, but it's got this efflux pump which pumps them back out. That's one way. Okay, maybe the antibiotics will come in and there's an enzyme that's being made now that will adjust the antibiotic so it's not going to affect them. Or maybe the antibiotic is going to be broken down by a new enzyme that plasmid DNA is calling for. So these are some of the ways that genes on the plasmid can help that bacteria become resistant. So how do we avoid it? We need to avoid taking antibiotics too much. Don't expect the doctors to prescribe them if what you have is really caused by a virus. When you are prescribed antibiotics, make sure you take them to completion. Otherwise, you could basically kill off most of the bacteria, but some of them are still there, and those can become resistant. You're going to kind of keep those that are more resistant to the antibiotic, and then they can reproduce being resistant to the antibiotic rather than killing them all completely at your first chance. Also, it's kind of implied, but make sure you use proper hygiene and other disease control methods. So that's some ways that we can help in the fight against antibiotic resistance. So the videos, I will link them to the website, and I'll include them at Modo, but they're good little videos that talk about antibiotics and antibiotic resistance and be worth watching. Alright, so that was Kingdom Bacteria. Now we're going to talk about Kingdom Archaea, and there's not a great deal that we're going to be responsible for them. Again, this is the newest kingdom. We used to talk of 5 kingdoms, but then they learned that those that were in what we called Kingdom Monera were really 2 groups. So it wasn't just bacteria. There's 2 separate groups that are very different from each other. So we have Kingdom Bacteria and Kingdom Archaea. Kingdom Archaea, also prokaryotes. So their cells are very small, simplistic, no nucleus. They have ribosomes, but no membrane-bound organelles. Very, very simple. The unique thing about the members of Kingdom Archaea is they can be found in extreme environments. So you can find them in volcanoes, you can find them in the seafloor vents, you can find them in the Dead Sea, which is like super, super salty. They look a lot like bacteria, and they can have very similar shapes and can be classified similarly based on shape, but they differ genetically and biochemically. So they have different DNA and proteins that give them very different properties. And their cell walls are very different too. Okay, so some of their characteristics, their cell walls, like we said, are not composed of peptidoglycan. And peptidoglycan is found in both gram-positive and gram-negative bacteria. Okay, so peptidoglycans are not in the cell walls of Archaea. Rather, they're made up of unique lipids or fatty substances that are super, super stable. That's why they can withstand extreme environments. Okay, they're super stable at high temperatures. So, along with that, they have enzymes in them. So these types of Archaea or Archaebacteria have enzymes in them that can remain stable at extremely high temperatures. For us, well, definitely for scientists, but in the long run for us too, these finding of these bacteria and the enzymes in them have been really, really important for advances in forensics and biotechnology and industry, because what we can do now is something called PCR, and PCR stands for polymerase chain reaction, and basically it's a set of steps or reactions that we can perform. That it's almost like a photocopier for DNA, so it allows us to make copies of the DNA. So if you think about a crime scene and you only have a tiny little sample of DNA that they found from the guilty person who committed the crime, if all that we could do is use that tiny little sample, then we can't do many tests or can't repeat them. But if we can make copies of the DNA and then have copies that we can use to perform the tests, then we can do a lot more analysis and be very thorough. So not only for forensics, but also for if you're studying different maybe really, really old species and you found a sample of DNA from them, or like any kind of studies with DNA, if all you have is a tiny bit, this allows you to make a whole lot of copies so that we can do more analysis of them and be more sure of the results. Okay, so they're really important for PCR. And PCR is a hugely important step in forensics and other types of biotechnology. Like I said before, Archaea, a lot of them have very similar shapes, but they also have other unique shapes as well. So there's some that are triangular, some that are square, lots of different shapes in the kingdom Bacteria and kingdom Archaea. And like those in kingdom Bacteria, they can also reproduce by binary fission or conjugation. Excuse me. There are 3 major groupings of Archaea that we'll be looking at or talking about. There are what we call methanogens. So there are hints right in the name. Okay, so the methanogens, they live in oxygen-free environments, so they live in anaerobic conditions, and the key thing about them is they produce methane, so that's why they're called methanogens. So methanogens would include, like, we have methanogens that live inside of the ruminant's stomach, so inside of a cow's stomach, and they produce methane. Halophiles, remember if we talked about hydrophilic, the philic part means to love, and then the first part tells you what they love. So hydrophilic means they love water. Halophiles, or halophilic Archaebacteria, love halo. Halo means salt. Okay, so halophiles love salt. They live in extremely salty environments. Now this part, we said thermo and acidophiles. Thermo means temperature, so they really like hot and acidic environments. So they live in extremely hot or acidic environments. Oh, and that ends kingdom Bacteria and kingdom Archaea. Okay, again, if you have any questions, I know there's a lot of information in these sections, so please, if you need any help, need some tricks for how to study them, have any questions about your assignment, just email or make arrangements to see me and we can work on it.